Yes, can I ask a question?
Yes.
So you said that it will be the final decision about joining INSDC will be released this year.
Mm-hmm.
So would it include all the details you mentioned, metrics, etc.? Because we were also discussing earlier. Would the detail in these metrics be released at this time?
No, actually, still we are discussing about what metrics is appropriate. Maybe if the metrics is too easy, so too many institutes will apply.
Right. Yeah.
It is. If it is too hard, anyone could be apply for the INSDC. So
right.
We are now thinking what is the appropriate metrics. Yeah, but we are now thinking requirements and metrics.
Mm-hmm. Mm-hmm. Thank you.
Hi, may I have a question? Okay, you mentioned that NGDs of China and COBIC of Korea are interested in joining INSDC. And do you know any other institutions or data centers that have interest in joining INSDC?
Actually, yes. Professor Ma said the IMCAS, one institute for the microbe in CAS from China, also want to join us. And maybe, of course, the Sintians BGI will apply. And for the Korea, I think maybe, Chris, KRISS. Do you know?
Yeah, Chris. Yeah.
Chris Chris have the COVID sequence data, and they are interested to join the INSDC, I think. So if the many institutes from one country comes to us, it is a messy situation. So, we are afraid for this. Yeah. Okay. So let's move to the maybe next one is Yiming. Please make your presentation.
Okay, thanks, yes. Let me share my screen. Okay, can you see the slides?
Yes.
Okay, good. Well, thank you, yes, and thank you, everyone, for this opportunity to introduce the progress of our center. We are a National Genomics Data Center, and also on top of that, we have CNCB, which stands for China National Center for Bioinformation. This is a new development. I believe I introduced this concept last year. So I'll just mention very briefly at the end of this some development. And I'll talk in three parts. First, some updates. So this is our team. So we have senior advisors, Professor Chen Renshen and Zhao Guoping, for us. And currently we have 6 professors now, which is actually 2 more from last year, because this year we have Professor Gao Yue and Shu Hui was promoted to for professor this year. And also we have 2 collaborators in CAS. And our overall team, we have 60-some students and 53 staff. So this is just show the growing of capability. So we start from 2016. We have 6 modules of data, kind of databases or resources. And over these years, we are gradually increasing different types to including many, many more. And we are kind of divided them into different areas. For example, raw data and others, and so on and so forth. So the data growth is still keep growing, and as for yesterday or as of today, our total data size is 18.7 petabyte. And so it's still very growing rapidly. So for each petabytes, the accumulation date from the first one from over 1,000 days to the recent as short as 30 days. So it's growing really fast. And also we are taking lots of data from many government or other grant supported projects. So this also shows the rapid growth. So the major grants are from most, the Ministry of Science and Technology and China's NSF and CAS, Chinese Academy of Sciences, and others. So you can see that especially this year, the growing is pretty fast. It's about 80% increase. And also the previous data size is also about 80% increase. And also our GSA data also support lots of publications. This show the growth, and currently it's close to 2,000 and covering 450, more than 450 journals. So the top one is Nature Communications. So, and also we have a separate database to host human data. So we call GSA Human, and also it's growing also very rapidly. And development of this is that we are currently we are taking up the human data backup system required by the Chinese Ministry of Science and Technology. So this was the previous protocol that submitters need to follow. So basically there are 2 routes. First of all, they need to back up their sequences to a backup center. And then once they get a backup ID, then they need to register at the most Ministry of Science and Technology Center to register. And once they got registration ID, then they can start the submission to databases like our GSA, and so that we can assign accession numbers, etc., so that they can do sequence search. Because one thing I should point out is that in this backup center, this is solely for backup. There is no way for people to search or retrieve data. So this is basically a black box. And so obviously, you cannot use this as evidence to the journals for publication. So in this case, in the old system, a submitter has to make 2 submissions if they want to publish this something or share this to the community. So we start to discuss this situation with most, and we finally last year we get rid of that part so that we combined the backup and the public database into one system, so that our center now take part in both functions, and so that this it's giving researchers a very simplified way to both meet the requirement of the government and also the need for publication and data sharing. And we also, in terms of other databases, our genome warehouse, GWH in short, which is for assembled genome, and also the data increase is very rapidly, especially in animal and plants sequences. And also we are developing multi-omics association knowledge bases. So we are now have GWAS Atlas, EWAS Atlas, and this year we add TWAS Atlas into our collection of these knowledge bases. And for resource for COVID-19, we continue to expand our data collection. So the total genome size is over 14 million sequences. And we also developed some new tools. For example, this one is a machine learning detection system for high-risk SARS-CoV-2 variants. So we are using various ways, different features to do this kind of detection. So the detection can actually shorten the warning period. For example, WHO assigned lineage like Omicron or Delta, etc. But usually there's a delay from the time these variants occurred to the time WHO assigned these names. But with our system, we can shorten this warning period. Something like so. This is an example of what we can we do. So and also we apply this to a web system so that I think we are doing update every 2 weeks. So we are it's a scoring system. So the higher the scores, the higher the risk is, so that you can tell that these are some of the newest occurring virus variant. So we are keep expanding the bioinformatics toolkit, BIT in short. So this year we add some more alignment tools in addition to the BLAST, localized BLAST I mentioned last year. We add some other alignment tools. So infrastructure-wise, we are continue expanding in both storage, application clusters, backup systems, internet bandwidth, high-performance computing, and high-performance storage. So these are the updates. So I'll briefly introduce some of the new developments. So one is the monkeypox virus resource, similar to the COVID-19 one. We established this very rapidly and released the resource in June. So it has similar function as COVID-19 resource. So basically, it's a collection of all the public genome data, mostly from INSDC. And also we have various functions from genome variation analysis to genome knowledge to literature and online tools. And another one is, as I mentioned, is a TWAS Atlas. this is going to publish in NER database issue. And also we developed cancer alternative splicing atlas. And because of the time limits, I'll just mention this. And if you're interested, you can refer to our website or the associated publications. And most of these will be published in the NAR issue, database issue this year. So this is a brain catalog, a one-stop for brain-related traits. And this is a cancer genome and consensus annotation system. And this one we call ProPan. It's a comprehensive database profiling and prokaryotic pangenome dynamics. And this is a homologous gene database, HGD, and it's also going to be published in NAR. And cell taxonomy. Okay, so these are pretty much the new development this year. And also development is that we are this year there's a call for the global core biodata resource selection run by Global Biodata Coalition, mostly initiated by institute in Europe, mainly EBI, etc. So they are doing some kind of selection for this core biodata resource. So we our GSA data applied for this and was initially selected, and we submitted the full application, and we are waiting for the final result now. And we also participate the INSDC annual meeting this year, as Yas mentioned. So we are very grateful for INSDC to allow us to participate their annual meeting to present our work and doing discussion. And one of the topic discussed this year is about NGDC's INSDC data exchange. And as Yas mentioned, there's INSDC is setting up the activity metrics for the inclusion of other databases into INSDC. And we are also having a discussion with other members of, individual members of INSDC. For example, this one with EBI to discuss database commons and the EBI search engine. And also, NCBI is very grateful to offer curation training for us earlier this year. So this is NCBI's team doing online training. They did 7 times this year for us. And because it's important for INSDC inclusion, it's important to do data mirroring. So so we realized this, so we started doing some data mirroring from biosample, bioproject, and SR data at from INSDC. So basically, we are currently, we've already take all the biosample and the bioproject data from INSDC and incorporate these data into our own system. And from April this year, we are downloading all the daily updated SRA data into and integrate them into our GSA. So, for example, if you go to our GSA database, you see 2 tabs for data. One is our own GSA data, and the other is the integrated INSDC data. So the data file currently is 1.6 petabytes. And later on, when we have time and resource, we'll going back to the historic data and try to catch up all the old data. So as I mentioned, we have integration of SIA data, and then we provide for these kind of data, we provide different ways for people to download these data. So for example, for NSDC data, we provide NCBI, EBI, and DDBJ download route. And also we provide our NGDC route so that people can, depend on where they are in the world, they can select the most appropriate route to download these sequences. And as I mentioned earlier, we established this gene-based database this year. And it's, as I mentioned, this equivalent to the GenBank database or ENA type or DDBJ type of database. So, so it's a database that can take submissions from researchers, and also we integrate the most recent GenBank release into the system, so that currently it has sequenced more than 233 million nucleotide sequence and 185 million protein sequences, covering close to 600,000 species. And since we just make it online, so we are also taking some direct submissions, 1,300 also nucleotide. So this is the system we are actually using to make it easier for later on exchange with INSDC. We are taking advantage of the GenBank FASTA and AS1 format, so that all our submission pipeline and display and everything are kind of mimic the GenBank style. So this is our system. And this is our submission portal, very similar to the Bankit system at NCBI. So people can just follow straightforward steps to fill in their to submit their information. So I should mention we actually kind of make two of the steps more kind of a controlled way. One is the source modified table. So for people who are familiar with this, these are basically all the metadata information, for example, organism name, collection date, country, etc. So we are doing this into a validation, a kind of a automated and kind of a smart system to do auto validation. And also to make it easier for people to submit, we are using the Excel structure to put all these fields and put all the instructions into the Excel table and have instruction in both English and Chinese, so that people can understand this. And also there's various validation steps. For example, we can do structure validator, column validator, row validator, cell validator, all kinds of different validations. So that once they have errors, we will clearly mark those errors, and so that submitters can fix them easily. And also for this is a kind of example of kind of errors that we provide. And also for feature tables, so these are the kind of table for annotation. So we provide three optional formats for submitting these feature tables. One is a plain text table or GFF3 format. So basically for large scale but also for small scale, people can also use the third option, which is the Excel-based table. So again, this is very similar to the source modify table. So we have very detailed structure, and we have all different pre-filled and controlled vocabulary. So to make to try to minimize the chance to introduce errors, and also there is a validations. So this is our search and download option interface. So you can do search and show similar result. And then you can display different formats from GenBank, accession list, ID list, etc. And also can sort by different ways and download in different formats. And so this is the display of the flat file, very similar. So as I said, we mirrored GenBank file. So if it's from GenBank, it looks like this, and with all different links to GenBank, original GenBank record. And this is our own record. So it looks very similar to what GenBank record looks like. So, so this is mostly our own work, and also we are doing lots of international collaboration, as I mentioned last year. So, one very good progress of this year is that we were awarded several grants for international collaboration. One is the continuation of support from IUBS on open biodiversity and health big data. So, and with collaboration of multiple countries. And the second one is a grant from a CAS for global genomics data sharing. Basically, this is the data mirroring and data sharing work that I mentioned with INSDC. And also we got two ANSO grant. ANSO is a international Society for Scientific Organizations. So one is for precision warning method for high-risk variants of emerging infectious diseases. This is in collaboration with institute from Brazil, France, and Pakistan. And also we have just this week we got announcement that we got another ANSO grant with Thailand and the USA for doing some TV work. So some very briefly, some future directions. So just a very brief progress report of the CNCB infrastructure. So as I mentioned, we got this CNCB title in 2019. And the government now is actually put a very great big project about 1 billion RMB to do infrastructure establishment for CNCB. So this is already past the initial application, and supposedly it's going to be starting to put the building next year sometime. So we're hoping that with this infrastructure, we are going to enhance our ability to handle all these submissions and the data sharing. And also we want to use this opportunity actually to collaborate with DDBJ and COBIC. And in this sense, also we also welcome any other institutes worldwide to actually our overall goal is actually to promote data sharing in whatever way, so that the scientific community can benefit from it. And also, as I mentioned, the global core biodata resource, we are still keep working with them to see I think this is a kind of a new system. Also try to improve data sharing. They're fine because as Yas actually mentioned that the rapid data growing, right? He used the SRA data as a good example. So I think this is kind of an example of how people everywhere trying to find a way to most efficiently share data. So we are also trying to seek all these opportunities. And of course, we'll still following up the partnership with INSDC. And we're very glad to hear from Yas that there will be some progress. And we really look forward to some further news to that. And one last thing is we urgently need talent. So we're still looking for talent to join our data center because, as you can see, we're doing lots of work and our data increase rapidly. So we need all different talents in all different aspects, whether it's an IT part or the curation side. So if anybody is interested in joining us, you're welcome to contact us. So lastly, I would like to acknowledge all the funding agencies and also, of course, the hard work of all staff and the students in our center. Thank you very much.
Yeah. Thank you for mention to collaboration with TDBJ and COVIC. It is important to make a good collaboration among us. And do you have any question or comments? Sophia?
Yes, thank you very much for this impressive talk and actually on the same time inspiring as well, I would say. The work provided, the work that you show is extremely interesting. I've got two specific comments. You mentioned, actually I missed the very beginning of your talk because of the time difference here, it's a bit early. You mentioned this collaboration with Thailand and the sequence of the whole genome of the Macaca fascicularis. We would be interested if you could share the immunoglobulin and the T cell receptors loci to do the annotation of this loci in order to incorporate the data in IMGT. That's one comment that we could discuss later on for potential collaboration. And then you mentioned about this, another specific question. You mentioned this gene base and that you have submission criteria. And I wanted to figure out, I have a really specific question. We have, for instance, extracted a sequence, a big sequence, a long sequence from NCBI. And then we have identified one gene that we are interested in. Is there a possibility to resubmit this sequence and have a precise accession number from gen base, for instance? We wanted to ask this question to NCBI or to EBI, but I'm taking advantage that you mentioned your database now to see if this is possible, because most of the times they're experimental data that they are needed in order for a submission to take place. In our case, IMGT, we extract publicly available data and we annotate them. Once we find a novel gene from a different assembly that we would like to give it an accession number, so it is recognized in the publicly available databases, and it is not only an IMGT number that it's not connected to the other databases. And my question again is it possible to do it via your database? Or we could discuss it later on if it's not clear to you. But in any way, thank you very much for everything.
Okay, thank you very much, Sophia, for coming. And Sophia is our BHVD member, and also she's one of the collaborators in one of the projects that we just awarded. So thanks for joining us. So about your two questions. So first, the monkey genome project. I think this is again with another partner of our BHBD Alliance. So I'll connect you with that party, and then of course we can discuss this later on to see if they can provide these immunoglobulin sequences later on to you. Okay. And as to your second question, I'll try to answer first, and then Yas should be more experienced, and Yas can correct me if I'm wrong. So my understanding is you want to extract a gene, right, from a very long sequence. So this sequence could cover maybe 20 genes or so, but you want to just extract that gene of your interest and assign a separate accession so that you can reference it, and it's easier for people to download this region, right? So okay, I got it. So my understanding is, so you said if we can resubmit it. So my understanding is there's a mechanism in INSDC called third-party annotation. I believe that belongs to that category. And I don't know if Yas agrees with me, because this is somebody else's sequence, and it's just that you re-annotate it and you want to submit it again. Yas, is that
Yes.
Okay? Yeah. So,
yes,
but
it is possible to add another annotation to gene-based NGDCs, but it would be make some problem if you would be merged when the NGDC become member of INSDC.
Yeah.
So therefore, it is better to use TPA, third-party annotation method of the INSDC, I think.
Right. But there is a condition to submit TPA, the third-party annotation, is that you first have to publish this work. So basically, you have to kind of say something. You already did some study or some research on these, and to make sure. But actually, it's not. In this sense, it's not that you are re-annotating them, because the coding region, for example, is exactly the same as in the long record, right? And the annotation, for example, the function, the name of the protein is exactly the same, right? You are not changing anything.
No, not really, because they are just nucleotide sequences, and there is annotation, there is identification of the immunoglobulin gene, for instance. So it was not known beforehand. And the publication, it's only in our web pages most of the times. It's not in our different releases of the data sets. It's not a publication in a journal.
No, so Sophia, it's not that you need other publication. For you to submit this, you need to publish something, so that it's make sure that your claim is valid. So what you say is scientifically reasonable. So
thank you.
Okay, thank you, Sophia.
Thanks a lot.
Okay, any other questions? Sengu? Yes, please.
Okay, so Yimin, I have a question on Global Biodata Coalition, GBC. So I am an observer member of GBC, and I'm aware that there's a selection for GBCR. And when you were submitting your application, were you given an option to apply as the GBC databases as a whole, or were you be able to apply only for a specific individual database that NGDC is running, such as GSA that you are operating now? So my question is, do you understand what I mean?
Yes. So yeah, thank you for this question. fully understand. My understanding is that GBC is solely for individual databases. It's not for data centers. So, for example, DDB, well, maybe I can use NCBI as an example. NCBI cannot apply for it. Only individual databases like GenBank or SRA can apply as GBC. So for us, NGDC cannot apply. We can only apply like GSA or our GWH, or only apply to individual databases. That's my understanding.
Okay.
Okay, so well, yeah, time is running, and so we'd like to move the next talk. Thank you, Min. And
thank you, everyone.
Well, the next speaker is Seung-woo from COBIC. Are you ready?
Yeah. 이거 어떻게? 아, 이거는 왜? 네. Is it close? Yeah.
Sure.
We have some technical difficulties. Please hang on.
쉐어 누르시면 되는데 이거
그건 아니에요. 아마 쓸 때는.
네네.
Hold on, we're almost done. Okay, can you see my screen? Right.
No.
Yes?
No.
No. Okay.
Oh.
제자리에서 큰게 접속한다. 그게 날지도 모르겠네.
Yes.
Oh, yes. Okay. Oh, sorry about that. So I'd like to give you
not a presentation, but your memorandum mode. Change the screen.
Okay.
Yes. It's okay.
Okay. Sorry
잘
about
보입니다.
that. Yes. Thank you. So I'd like to give you update report on COBIC and mostly, mostly Korea Biodata Station, KBDS. My name is Sung Hwang from Korea Bioinformation Center. So, okay, let me begin by introducing Korea Biodata Station, KBDS project. The goal is to collect all types of biological data. So we are not just talking about omics data, but every type of data that were produced from all Korean government funded research projects. And we want to act as a data repository for both national funders and international publishers as well. And the KBDS website opened last year, actually, but its official version will open next week, next Wednesday, after a year of complete revamp. And the question is, why COBIC collects data from Korea? Well, for scientific reason, it's obvious, to enable data-driven research, right? And for policy reasons, it is COBIC's duty given from the government to collect data. And there are two laws: Act on the Acquisition, Management, and Utilization of Biological Research Resources. And in Japanese and Chinese characters, the law is as follows. And there is also National Research and Development Innovation Act. So under these laws, COBIC must collect biodata, and Korean researchers are required to submit their data to national repository. That's the reason why COBIC collects data from Korea. And why did COBIC develop KBDS, a new data repository? For policy reason, as I just told you, Korean funding agencies are requiring researchers to submit their biodata to national repository. And also, sensitive controlled access human data should be handled in such a way that complies with relevant Korean laws and policies. And for technical and other reasons, we need a repository that can accept submission of all types of data, not just omics data. And a variety of nationwide large-scale projects are underway. So their data must be deposited somewhere. And there is also a network bandwidth problem. If a Korean researcher wants to deposit to or download from NCBI, the speed will be on the order of 100 megabyte per second, whereas if within Korea, it will be 9 times faster. So that's the reason why we developed KBDS, a new data repository. And currently, the major driving force for data submission in Korea to KBDS is Data Management Policy, or DMP, in Korea. And the DMP is laid out in the standard guideline on collection, management, and utilization of biological research data. And under this guideline, researchers need to submit their data in each year as well as upon completion of research grant. And they can ask for extension of the data release for up to 1.5 years to funding agencies. If funding agencies approve, then COBIC will extend the data release. And this guideline is currently applied to only a limited type of projects, such as large-scale data generation projects and a specific project called Biomedicine Technology Development Project. But this data management policy will be strengthened and will be applied to a wider range of projects after law amending, which is currently underway. So to collect all types of biological data with comprehensive context information, we need if de facto international standards exist, we can benchmark them as much as possible. And the process was community driven. The development of data submission forms was not done solely by COBIC, but it was developed by several committees of more than 100 researchers. And The bioproject and biosample data submission forms were based on INSTC's forms with some modifications, such as removing some fields, adding some fields, and modifying some fields. And there are several types of major data, and the submission forms of major data types were also developed based on other existing forms, such as for NGS read, it is based on NCBI SRA form. For microarray, it is NCBI GEO. For nucleotide, it's NCBI GenBank. For proteomics, it's EBI Pride. For metabolomics, it is EBI's Metabolize. For chemical, it's NCBI PubChem BioAssay form. And for bioimaging data, it is DICOM, BIS, IDR, EBI Bioimage, and many others. And for these submission forms, we did some modifications also. Okay, but there are many biological data other than these standardized and very well-defined omics data or chemical data or bioimage data. And they are called collectively as a generalist data or small science data or orphan data, meaning that those type of data do not have a specific dedicated data repository to go. So we benchmarked 6 such generalist repositories, including EBI Biostudies, Dryad, Figshare, Zenodo, Mendeley Data, and OSF. And we came up with a simple data submission form for other unstructured data, including consisting of keywords, additional notes, file upload, and in many cases, they are just Excel files and the description of files. And if the submitter has already submitted, what's that? Oh, okay. Links to external databases and description of links. And so I think I predict that most of the data in terms of with respect to submission number, the generalist data or will account for most of the submission number, whereas with respect to submitted file size, genomics, proteomics, metabolomics, chemical, and bioimaging data will take the majority. And we also took one step further and developed additional forms for miscellaneous data. Okay, so we are covering all these data, but the problem is that COBIC has expertise only in genomics. So how can we make sure that all these proteomics data, metabolomics data, chemical data, and bioimaging data are of high quality? Well, we outsourced them. So we established Center for Data Curation and Quality Management Center. They are virtual centers. So we appointed for each data type, we appointed virtual centers external. So for example, for genomics data type, COBIC is the center that will be responsible for the quality assurance of genomics data. But for proteomics, Korea University is responsible for QC of proteomics. And for metabolomics, Seoul National University is the one. And for chemical data, it is Korea Research Institute of Chemical Technology will be the virtual center for the chemical data curation and QC. For bioimaging data, it will be Ewha Women's University. So that's how we make sure that each type of data will be collected in a high quality form. And each of the center has plans for international collaboration. For us, genomics part, for us, of course, we will continue collaboration with ABC, DDBJ, and GDC, and as well as NCBI and EBI. For proteomics, they are planning to make collaboration, including HUPO for metabolomics, Massive, and the Metabolomics Society, and among others. For chemical, they are planning to collaborate with EBI Campbell, etc. For bioimaging, they have plans to collaborate with EBI, PDB, LONI, Allen Institute, and etc. And we also established Human Data Bank from the Ministry of Health and Welfare. And we also made the provision for the operation and management of Human Data Bank. And we also established the Data Access Committee so that when there's an application for data access of controlled access human data, Data Access Committee will review it. And if they approve, then the applicant can download the controlled access data. And we recently, last month, we submitted 2 manuscripts. The first one was KBDS, the manuscript about KBDS. It was submitted to Genomics and Informatics. The other was KONA, Korean Nucleotide Archive. What it is that it's a subsystem of KBDS for dedicated to sequence read data. And it was submitted to Genomics, Proteomics, and Bioinformatics. So for summary, KBDS opens just next week. And KBDS accepts submission of all types of biological data. And the major driving force for KBDS submission is data management policy in Korea. And we are equipped with a variety of data submission forms. And we are ready for dealing with controlled access human data. And we are eager to bring our collaboration to the next level more than ever. Thank you.
Thank you for your presentation, and well, congratulations of the great progress of the COVID. Any questions? So from me, well, you said you add some modifications to the bioproject and biosample in COVID, but well, as I mentioned, if you are considering getting the INSDC membership, I'm concerned that you may have a problem if you do not get the exact match with the INSDC's structure of the bioproject and biosample.
Yes. So for that matter, we will make sure that our forms have exact compatibility with the existing INSDC form, meaning that many of the modifications we have to revert back before modification. We will do it certainly.
Thank you. Imin?
Yes, thank you, Yas. And thank you, Suwon, for your wonderful progress report. This is very impressive. And follow up with Yas' comment, actually, we actually had the same issue early on when dealing with bioproject and biosample with INSDC. And we also have inconsistency in, for example, in the different field, we have some extra ones and we're missing some ones, something. So we are also kind of doing this kind of compatibility and doing some kind of matching. So it's kind of natural. I believe, you know, each center when they first started, they have their own. So this is, but we have to kind of deal with it and kind of, as Yas mentioned, have to compile, kind of to meet the INSDC requirement. And my question is actually for your mentioning that you kind of outsource different type of a database to virtual centers. This is actually a very good system to deal with this. So because we are also kind of doing something very similarly. So my question is, what kind of, how do you, or what's the mechanism of this collaboration? For example, who's paying for all this work? Do the government paying for different virtual centers, or you are kind of like have subcontract to these virtual centers? And the second question is how you kind of interconnect all these different data centers. For example, we kind of have to have sort of cross-referencing to different data type. So is this something that you as a I would say core center is doing, or how is dealing with? Thank you.
Okay, thank you for your question. So for the first one, it is government who pays the grant to the virtual centers. Mm-hmm. They are not paid by subcontract of our grant. So it is government, not directly from us. The grant source of money is from government, not from us. And the second one, all the databases that in the KBDS is just one huge big database as far as I know. So the information should be integrated there. And for all the other, except for the chemical data, all the metabolomics, proteomics, genomics, bioimage, they use the same biosample database. So they should be they should have links to each other by way of biosample record. And as far as I know, it's a very big single database, I think. Yeah. So did I answer your second question?
Okay, so yes or no. So basically, are the data in different virtual centers physically? So if you are saying that it's a one big database, mm-hmm. then all these data, although they are doing the curation and collection, and they are doing like acting as a domain expert, but actually the data themselves are actually all stored in your center. Is that how it works?
So all the hardware, all the software, all the data, all the databases will be operated by COBIC.
I see. Okay. Right. Understand. Okay. Thank you.
Any other questions? Okay, so if not, thank you. And we'd like to have a break until 4 p.m. in Japan and Korea, and for China until 3 p.m. Okay. I'm sorry. Let's start next session. The first speaker is Yasuhiro Tanizawa from National Institute of Genetics and DDBJ. And his title of talk is sharing the genomic data in the post-model organism era. And so please start, Tanizawa-san.
Okay, can you see my desktop?
Yes.
Okay, so let me start my presentation. So today I will give you a talk with this title, Sharing the Genomic Data in the Postmodern Organism Era. I'm from National Institute of Genetics. My name is Yasuro Tanizab. I'm very happy to have a chance to talk here. Before starting my presentation, I would like to show you some of the works in our laboratory from microalgae like diatom to vertebrate. We have various kinds of genome projects. In this project, we have assembled the genome sequence and conduct genome annotation, and sometimes we develop a genome database to share genome data among collaborators. Later, I will talk more in depth about the liverwort mercantile polymorpha, which is now attracting interest as a new generation model plant. Also, I am working on prokaryotic genome projects. Working together with these partners, I have so far conducted taxonomic studies of lactic acid bacteria. This one shows some of the examples in which I have proposed a new taxa of lactic acid bacteria. And these studies are unique in the sense that they are proposed mainly based on the bioinformatic analysis using publicly available genome data rather than experimental works. In addition, I am developing a genome annotation pipeline called DFAST. It stands for DDBJ Fast Annotation and Submission Tool. As its name suggested, it runs very fast. It can annotate typical size bacterial genome within 5 minutes. And it can also generate data submission files that can be readily registered in DDBJ. Today, I don't have enough time to talk about DFAST, but if you have interest in this, please try this from the URL shown here. Okay, let's go back to the main title. And as you know, the genome sequencing technology is rapidly advancing. We can now easily obtain genome sequences in a shorter time with less money. This means that genome projects can be conducted by a smaller research group. In other words, the scale of each genome project is shrinking. While I was involved in many genome projects, I came up with an idea that there is a growing need for data sharing among a smaller number of collaborators. I will present my talk by separating it into these two subtopics. First, I will talk about mercantile as an example for the data sharing of a model organism. Second, I will talk about data sharing that I think is suitable for non-model organisms. Actually, this is not about what I have done, rather it is about what I am trying to do or what I want to realize in the future. But anyway, let me start with the first topic. The liverwort mercantile polymorpha is considered a basal lineage of plant plants. While it retains many orthologous genes that are common to other land plants, the genome analysis revealed it has low genomic redundancy with fewer repetitive sequences. Further, it is easy to handle in the laboratory, and while variety of genetic tools and techniques are already established. Importantly, the haploid phase is dominant in their life cycle. The pictures shown here are mature plant bodies for male and females. They are in haploid phase. So therefore, it is relatively easy to observe the phenotype as an effect of gene knockout or knockdown. For these reasons, more and more people are using them as a model plant. The latest genome sequences were released in 2021 with its total length of 223 megabases. And every 2 years since 2010, the mercantile workshop had been held, although it is cancelled because of the pandemic for the 2 or 3 years. The history of mercantile genome project is shown here. Although the organella genome sequences were determined in a very old time, but it was in 2070 that the reference genome sequence was officially released. It took about 10 years since the beginning of the genome sequencing project. But soon after the genome paper published, we have a new chromosome level assembly. It was presented at the mercantile workshop in 2017. I joined this project to conduct genome annotation, and it was released in 2020. So now the latest genome sequence is version 6.1, which is a combination of the version 5.1 male genome and female sex chromosome sequences. But during this period, I hosted the genome annotation jamboree, where mercantile researchers joined the curation of gene annotation. So I will talk about this jamboree in the next slide. The genome annotation jamboree was held during the mercantile workshop 2090, which was held in 2090 in Sendai, Japan. It was a one-day event, but it was not enough to finish the work, so I assigned a task to each participant and asked them to finish their tasks by the end of October. There were 68 participants, but in total, we created more than 2,000 gene models. So these pictures showed the mercantile workshop. I really appreciate the two people who helped me a lot during this jamboree. So this slide shows what we did in the jamboree. We collected the gene models using WebUpload, which is an online annotation editor. By comparing with the full-length transcript sequence from PacBio-ISEQ, or expression pattern based on RNA-seq, or cage signals, which indicates the transcription status type. The gene models were corrected to make them consistent with these experimental evidence. So this shows an example of the improvement in gene annotation. So in the previous version of the genome sequence, this gene contained falsely duplicated region shown here. Probably this was due to misassembly. But in the new genome assembly, it was corrected and verified using ISEQ full-length transcript sequence. After these works, I renewed the website for mercantile genome. Initially, it was launched as a supporting website for drafting the version 3.1 genome paper. And when the chromosome level assembly became available, I updated the website to store the new reference genome and name it as MapleBase. So it is a comprehensive genome database with genome browser sequence and keyword systems and various utility tools such as gene finder for extracting gene sequences from the genome, or just finder for finding target sites for genome editing. Now we have genome sequences stored in our databases. In addition to the reference genome obtained from subspecies ruderalis, which is the most widely used as a model, we have two other genomes from different subspecies. They were contributed by third parties. I added this data by the request from them. Our database has unique features as a database for a model organism. I will show some of the examples. One is our gene nomenclature registration system. Since many people use this brand in their research, it may cause confusion if they name their gene arbitrarily as they like. To avoid such confusion, consistent gene nomenclature system should be maintained. For mercantile, the naming guideline was published. This system helps in maintain the nomenclature system following the guideline. Now, it is recommended that gene symbols and names be registered before publications and presentations. It has about 3,300 names already registered in this database. Another unique feature as a database for model organisms is a gene identifier system. Since it can be referred in many papers, the system should be simple and easily understood. This new gene ID system was introduced as of version 5.1 genome, which is the first chromosome level assembly. This is very similar to that of AGI and that of Arabidopsis naming Arabidopsis gene identifier code. Because many plant researchers are already familiar with it. So, for like many other model organisms, it is possible that the reference genome is updated in the future. In such case, these gene IDs will be carried over to the new reference genome. And I also made a gene ID converter, which can convert gene IDs between different versions of genomes. And lastly, I will introduce a new database called MapleBase Expression Database, or MBEX. It was developed by Shogo Kawamura, who is a PhD student in Kyoto University. We contrasted this by reanalyzing publicly available RNA-seq data from the SRA low-read sequencing database. In total, we have 117 datasets obtained from different sample conditions. It was developed as an all-in-one platform for visualization and analysis. For example, users can visualize co-expressed genes as a network graph and then conduct functional analysis. And the results can be compared using a tool shown here. And MBEX also provides expression level images, which is embedded in the macro-based gene detail page. It is quite useful. I have several update plans regarding this database. New tools for helping artificial microRNA design, or I also have a plan to add gene family viewer, or a plan to incorporate more variant data. So, okay, so let's move on to the next topic. Okay, and as I already told, we can obtain genome data more easily than before. Now, any species can be a subject of a genome project. I will show you such an example from our recently published paper. We determined a nearly complete chromosome level assembly for red perilla. It is famous for the commercial food seasoning named yukari. I'd like to emphasize that it was mainly conducted by the two people, Dr. Keita Tamura in Hiroshima University and Dr. Mika Sakamoto in our laboratory. They finished this work in about 1 year. Of course, this establishment is attributable to their hard work, but in addition to it, it is also thanks to the state-of-art technologies such as PacBio, HiFi, or Omnisys scaffolding. Our genome is now designated as reference in the NCBI database, although there is another genome previously reported. As demonstrated in this example, we can obtain high-quality genome data with lower cost and with less human power. The difference between model and non-model organisms can be summarized like this. Needless to say, the research community is large for model organisms, while for non-model organisms, the genome projects are often conducted in a small group or even by a single laboratory. In terms of the quality of genome sequences, the genome of non-model organisms is comparable to those of model organisms. That is, as demonstrated by in the previous slide, it is achieved by the advancement of sequence technology. As for genome annotation, it may depend on the reference data. If the reference data from closely related species are available, the quality can be better even for non-model organisms. And in terms of genome databases, most of well-known organisms have their dedicated genome databases. MGI for mouse, SGD for budding yeast, TEAR for Arabic twisted, and of course, we have MapleBase for the liverwort. Developing and maintaining these dedicated genome databases are costly, and it may not be suitable for non-model organisms. Therefore, there is a growing need for a simple genome database that is suitable for data sharing among a small research group. I made a platform for this by exploiting the function from MapleBase. And by using container virtualization technologies, which realize portable and reproducible computational environment, the genome database can be launched without installing required software. So in this platform, the genome annotation data described in a standard format annotation file, such as GFF, can be readily imported. And such system, gene detail page, or utility tools become available. For BLAST web server, I used a third-party software called Sequence Server. It is also provided as a Docker container. The BLAST web server can be launched with a one-line command like this. Also, the genome browser is available as a Docker container too. If you install this this genome browser by yourself, it will probably take at least one whole day. But using container virtualization technology, you can make this genome browser in a minute with a command shown here. I developed a genome database for a domestic cat using this platform. When I launched this database, it was just before the press release and we did not have much time. But I was able to build this in a day or two. And also, we are developing the genome databases for red panda peach and Antarctic fungi and so on. And finally, let me summarize my talk. There is a growing need for data sharing with lower costs, with less human power, and in a simpler and more timely manner. So I made a genome database platform for small-scale genome projects, which is featured with essential functions such as genome browser, search systems, or utility tools. It can construct kits very easily in a short time with container virtualization technologies. Currently, it is still limited to internal use in our projects, because it is not well streamlined yet, and it is difficult to build this without my help. So the system should be more streamlined before it can be used by other people. And as a future plan, I would like to make this read and write database, which means users not only browse their data in the database, but also can revise or edit the gene annotation on this system. And in addition, users can submit their genome data to DDBJ. And that is my future plan. And thank you for listening. And that's all for my presentation.
Thank you very much. And so, Sophia.
Thank you very much for this interesting talk. You mentioned about this assembly, and I wanted to know if you have, if you could elaborate and you could give us a bit more information about possible assembly errors. Maybe I could be a bit more specific. We are currently working on different human genome assemblies and on this IG and TR loci, and several times where we are faced with the issue of deletions or even inversions in a different assembly. And I don't know if you have more experience on this and you could say what is the percentage of this assembly false duplications, as you say, or these things on the human genome. Thank you very much.
You mean this slide? Okay, so the previous version of the genome sequence was mainly constructed using short-read sequencers. So the newer version was assembled using the long-read sequencing. So these in the previous version, there are many false duplicated regions like this, and they were corrected when we used the long-read sequencers. So I found several of these. I found another examples like this, but all of them were correct in the newer assembly. And another example. So this shows the regions of highly repetitive sequences. In this region, there are at least 4 copies of genes annotated as metalloprotease. These genes were collapsed or fragmented in the previous assembly, but in the newer assembly using the long-read sequencer, they were assembled correctly, and there seem to be at least 5 repetitive regions.
Sure. What is your standard to say that this is a correct assembly then? How can you say that this is incorrect and the other is a correct assembly? What is your reference there, please?
Yeah, in this case, we have our ISIC full-length transcript sequence. It is a very powerful tool, and by comparing the gene models with the ISIC, we can verify it is correct. So without ISIC, it may be difficult to say it is correct or not. So is that okay?
Sure. Thank you very much.
Okay. Any other questions?
So I have a question. When you try to annotate a new genome as a non-model organism, do you have to do starting from the very beginning, like manually annotate some of the genes? Then
sorry, I can't catch your voice clearly. So please say it again loudly.
So when you try to annotate a new organism, do you have to start from the very beginning, like manually annotated the genes, then start to make the prediction? You mentioned that you manually annotated like 2,000 something genes. Is this step unavoidable?
In general, we transfer annotation from closely related species, but it is not sufficient. We have to conduct de novo predictions, and somehow we merge the results from de novo prediction and gene transfer from related species. I think so. This task is difficult to be automated, and some of the parts should be manually created.
Okay. Have you compared the annotation purely based on automatic pipeline and the result you manually created plus at the initial prediction? What's the difference?
Sorry, could you repeat?
What's the improvement you got by manually created some genes first?
Improvements of manual creation.
Right.
So many of the gene prediction tools can predict coding sequences, but transcription site is difficult to determine. By manually checking, we can add UTR to gene models.
Okay.
So example is shown here. The gene models shown this is lacks the transcription site. It is confirmed by comparing with amnestic expression data or cage data.
So, well, it's almost time, and so I'd like to move the next speaker. Thank you, Tanizawa-san.
Thank you for listening.
Okay, so the next speaker is Professor Wei from the Shanghai Jiao Tong University. Are you ready for?
Yes.
Yes.
Can you see my?
Yes.
Okay. Thanks for inviting me to present our work. My name is Chaken Wei from Shanghai Jiao Tong University. I will talk about the pangenomic analysis of Chinese gastric cancer. First, I will start with traditional genomic analysis or disease genomic analysis diagram, and which use the reference genome as the base. Then I will mention that the human genome is incomplete. Then why we use the pangenome and the pangenomics analysis. Then I will introduce our work on the Chinese gastric pangenome analysis. So as we all know, the Human Genome Project constructed a reference human genome. And after that, most of the biomedical study based on the human reference genome created that. So most disease genomics analysis will be something like this, by sequencing, then by sequencing the read and do the DHK like analysis pipeline to call the variation, then do like a genome-wise analysis, then verify the function of the significant associated genes, then go to the translation applications. Now there are many disease genomics projects like the cancer genomics and the TDD pan-cancer, or the pan-cancer care is not, well, it's similar to the pan-genome concept, but it's more emphasized on a different type of cancers here. And in my presentation, the pan-genome is only for one cancer, one type of cancer, but with many individuals. So as I mentioned, although there are many projects with using the human genome almost all of them now use the human reference genome as the base. So when you align the sequences, the sequencing data to the human reference genome, there will always be some, like 1% to 5% or more sequences, and they will be cannot be aligned to the human reference genome. So we call those a non-reference genome sequence. They were ignored. That's for almost all of the studies now. So this is based on the assumption that the human genome is complete. However, as I said, the human reference genome is incomplete. One reason is the sequencing error. Two is assembly error, and the other is the sample in when in the human genome project, different populations are some of the populations are underrepresented, and even after more than 20 years after the first reference genome completed, it's the first full genome sequence was only published in last year. So in this one, they create called a T2T, like terminal genome that without gap. This is the first computed human genome, only published in last year. Compared to the reference genome, it's not compared to the 20 years ago. It's compared to the current version of the human reference genome. The full-length genome can be 150 base pairs more sequence more than the human reference genome. So when I say the population representation in the human genome, this is the original human genome project. The composition of the samples, the ancestral source, and you can see because this is the human genome project is like a mosaic composition, not combining the whole sequence of the 20 or 21 individuals in the human genome project. It's like I picked like 65% of the genome coming from one individual, and different percent from the other individuals. But in terms of the ancestral source, like East Asia is only 6% in the human reference genome. So you can imagine that some of the region that might be more frequent or East Asia specific region will be, a lot of them will be missing in the reference genome. And also if you look at the GIS project, of course in other projects, like the number of samples from East Asia was relatively underrepresented. But if we look at the human genome data from different populations, when you sample more individuals, here is a review done in 2 years ago, that it shows that the novel sequence, like the novel sequence is like about 1% to 10% the size of the human genome. That's very different, or with relatively low identity percentage with the reference genome. Or I would say it's missing in the reference genome. In Han Chinese, this was done by my group, that we found about 29.5 million bases was missing in the human reference genome, which is about 1%. And in the African Pangenome project, based on the 910 African individuals, they found about 300 million base pairs missing in the human reference. So that's why I say about 1% to 10% of the human reference genome are missing. The size of the human reference genome are missing, and the problem is those missing or non-reference genomic sequence. Sometimes they can be very important. Like in this in the Iceland genome sequence project, they found this 766 base pair long non-reference sequence. It's associated with heart attack. So it has important functions. So we need to pay attention to those non-reference genomic sequence. Now in US and Europe, there are a trend to try to make a relatively complete or comprehensive reference for the human genome. So it's a human pan reference sequence. I will come back to the pan genome very quick. Anyway, this year, April, in Nature, one paper was published to try to promote to create a pan reference genome for the human, so that we can, like the human reference genome, it can provide a much comprehensive reference for all human genome related researches. So next I will introduce the pangenome and the pangenomics. This concept of pangenome was first introduced in 2005 on the bacterial genome. For one species, I think it's Streptococcus, when they sequence many individual or strains of this species, then they found many more genes, and they then come with this concept called pangenome, which is compared to the individual genome. For pangenome, those shared by all individuals were called core genome, and those show in some individual but not all are called dispensable. Well, this word dispensable means a little bit misleading. I like to call it a distributed genome that's shared by some but not all. Some of the region is only owned by one individual, not others. So these were part of the dispensable genome, but we call them a unique genome. So this kind of concept can provide more information about the genome contents. In bacteria, when they sequence many more individuals, they found that the number of genes increase. Like we will expect that they go flat when you have more individual samples. But at that time, when they sequence like 17 or 12, the number of genes increase. As you can see, there's new genes increase. They never goes to zero. It's going to around like 38 or 32. I don't remember exact number. It will like if you sequence more, the total number of genes will be like infinite number. So they call it open pangenome. So this is very interesting phenomenon but it's in bacteria, right? So we were very interested to answer that. What about the human pangenome? If we sequence many more human genomes, what will be the size of the human genome? How many genes will we have? But at that time, like 12 years ago, we don't have so many human genomes, high quality human genomes available. But we do have some rice genomes available at that time. So in my lab, we developed a software called UPen. It's a pangenome construction and analysis tool for eukaryotes compared to the prokaryotes, the bacteria. So it works for the rice genome. So we applied this for 3,000 rice genomes and created this rice pan genome. Interestingly, the rice pan genome, the size of rice reference genome is about 380 million base pairs, less than about 400. And when we combine the 3,000 rice genomes, sequence genomes together, we get about 270 million base pairs more, which is 70% larger than the reference genome. So this is very surprising that time, that a genome of species can be much larger than a genome of an individual, even for eukaryotes. And also, as I mentioned, that there are core genes and distributed or dispensable genes and unique genes. So the question is, in terms of function, can the dispensable gene be important or not? So we checked about 130 genes that's important in economically, and then we found about 20% of them are actually dispensable. So important genes can also be dispensable. I think we answered a lot of questions in the rice science, and also answered a lot of mysteries in rice breeding. Their questions. So come back to the human pendulum. What will it be if we construct a human pendulum? So my lab, we have those UPenn tool, but that tool requires very high memory. In our university, the largest computing node with memory that time was like 256 gig. But for the human genome, using that time we use super de novo for the genome assembly, requires 1.5 terabytes of memory. So we spent 1.5 years to replace the genome assembly tool with this GSA, which use a compressed data structure that will save about 10 times memory. But the speed will be like 7 times slower than the previous version. So, but anyway, we can use this new pipeline, finish the pendulum construction in our supercomputer center. Then we created this called Han Chinese pendulum using 275 individual genomes. And we got 33 million base pairs novel sequence. But of this 33.58 million bases, there are about 4.08 million bases were actually can be aligned to the Apache sequences in the human reference genome, GRCH38. When we compare these novel sequence to the other genome pangenomes from other population, like the African pangenome, then you can see like the African pangenome, they can occupy about 44% of our novel sequence. So these sequences are novel or non-reference compared to the reference genome. They are novel, but they are actually in the other population, like the African population. So the only about 40% of this 33 million base pair are like Chinese, the Han Chinese specific region. And well, other than the UPenn, we call it the UPenn 2.0. Now we we just published one paper early this year, we internally we call UPenn 3.0, which deal with the long read sequence, the third generation sequencing data that we found that the rice genome can be much bigger than we previously estimated, like 270 million base pairs more. Now we have like 600 million base pairs more new sequence when use the novel sequence. When we use the third generation sequencing data. So come back to the human pangenome. So we found this about 30 million base pairs new sequences in the human genome, and the question is what's that indicate for disease genomics? So because most of the data, 185 individuals were actually from the cancer patients. So we do have the tumor and the normal tissues data. So we focus on the tumor sample data. We get this pangenomic analysis of this Chinese gastric cancer. We have the normal tissue as 30x coverage, and the tumor is 60x coverage. The total data is 50 terabytes, and we have 10 phenotypes. Here is what we did for the pangenomic analysis. So first, we create a gastric pan reference genome, which include the human reference genome plus the unaligned regions. The unaligned regions have two parts. One is fully unaligned, another part is partially unaligned. Then we created this a pangenome for gastric cancer, then align the sequence again to this pangenome, use this pangenome as the base. Then we have the gene presents absence variation for genes in the reference genome, and for new genes. Those new genes will predict on those unaligned regions. Then we found those some of the gene presence absent in the reference genome, as well as some new gene that can have some function in the cancer cell growth. So we do have more new sequence unaligned. Well, the previous, I mentioned the 29.5 million bases, it was only for the fully unaligned. So after we consider partially unaligned region, the total size will be 80 million base pairs. Then the distribution of those partially unaligned regions looks, there's no specific rules. We cannot see any specific laws that we have some. We just show the distribution here. But if we use those GC-PAN, the gastric cancer pangenome as a reference, compared to use the human reference genome, the GC-RCH38 as a pangenome, you can see that the mapping rate of the sequencing data is increased significantly, both in the normal and the tumor data. And after we constructed those pangenome, we have 261 we call distributed genes, which is not shared by all individuals. Among this, 186 was shared by the normal and the cancer tissues. There are 14 novel sequences. Well, at the very beginning, we have many more, like 82 novel genes, but most of them have very high percentage of repeats. So we removed the genes with high percent of repeats. Then we get 14. Of those 14 novel genes, 5 are core, which means all those 185 individuals have this gene. and 9 are distributed. Some of them have them, some of them not. For the genes presence or absence, dispensable genes in the human reference genome, we found this is 186 genes. We compared their frequency in the Chinese population, in this cancer patient population, versus in the SGDP, the Simon Genome Data Project. We downloaded 263. We consider them as a healthy control sample. So compared to this SGDP data set, the cancer patients, the frequency of the gene presence absence frequency, those with orange color are those genes are significant. The difference, the frequency difference in the two populations are significant. So you can see there are many genes actually have very significant different frequency in different populations. So I list the 4 most significant different genes here. Also, these genes are they are like if you look at these lines, they are like they are very the CDS coverage. The color here are the CDS coverage, which means at least 50% of the CDS were missing for these 4 genes. So they are severely absent for each one, and also have very high frequency in the population that's missing in this data. The gene expression level, we have about 90 samples with a gene expression, and 67 of them are the same sample as the genomic data. So we listed the gene expression level here. I will say for those novel genes, well, here all these genes are from the reference genome. For the novel genes, the expression level will be much lower. Again, for that 4 highly absent and also very high frequent missing in the population, as you can see here, these 4 genes we call the high absent genes. The CDS coverage can be very low for some of the population, some of the individual, and because here is like 50%. Like you can see, many populations, many individuals have very low CDS coverage for these 4 genes, but for the others, because we use a threshold of 90% as the threshold to decide if this gene is missing or not. So at least for these 4 very severely absent genes, they may have some information for us. So we checked we show the real data for the frequency in different population. As you can see, in the so this gene ACOT1 is missing in more than 70% of the Chinese population, but in non-Asia population, only 23% is missing. And for the GSTM1 in this Chinese population, the gastric cancer population, also Chinese population, is about 60%, but in the non-Asia population is 46%. And most significant different one is here, the SIGLET14 in Chinese population or East Asia population is like 40% or more, or about 40%, but in the non-Asia percent, it's less than 5%. So these genes, their absence frequency might be a genetic explanation for the high rate of gastric cancer in East Asia population or Chinese population, because before people think the high rate of gastric cancer in China or in East Asia was related to our food habit or other more like not non-genetic issue, but now we do show some genetic can explain the high rate of gastric cancer. But for the UGT1, it is similar here. Here show how those what when we say missing, what that does mean in the genome size. And also here is when we say missing, sometimes it's the whole gene was missing. Sometimes it's like for here the HCOT1 is just 2 exon missing, 1 is actually there. And we did some function analysis by pathway analysis. Then like the 4 most most severely missing gene, 2 of them actually in the chemical carcinogenesis pathway. So that means the genes we found does have some meaning in cancer. Back to the non-reference gene, because we use the pangenome, we have the reference gene genome, and those belong to the non-reference genome. The 14 genes we use the transcript data to validate it, and I think most of them can be validated with pretty good data support them. So we start from 82 novel genes, and by the repetitive sequence contents less than 50%, we only have 14. Well, those genes with high repetitive region doesn't mean they are not real, right? But we just focus on those with less repetitive regions for the 14 ones. And the 9 distributed genes, 9 local novel genes, and we found, we use 7 long read 3-generation sequencing data from public data to try to locate those 82 novel genes. And we found that 9 can be located to the chromosome. Then together we only have 1 novel genes that is located in the chromosome 9. And as I mentioned, the expression level was relatively low compared to the genes in the reference genome. And also the 9 normal genes, 5 of them have very different frequency in the 2 different populations. The only one that's novel can be located in chromosome 9. Actually, it has proteomic data support that this is a protein coding gene. So we found at least 2 individuals in the proteomic data. We verify that this gene has peptide products. It's like 32 amino acid long. It's not very short, but I think it's pretty reliable evidence. And this new gene, we try to find what kind of function, because only this is a new gene and it's not showing all of the individual. It's only in 10%, like 20, less than 20 individual of the 185. About 10% of the population will have this gene. And we verified the function of this gene in gastric cancer cell line by increase the expression level of this gene. And you can see when we increase the expression level, the growth of the cancer cell will be reduced. So this gene can inhibit the growth of the cancer genes. Well, so far we starting from the cancer as a pan-genomic method applied to the cancer, the gastric cancer. Then the question is this method can this method be applied to other disease? So we compare this method and the traditional SV analysis method. If we only consider the heterozygous SVs, then the number of the variation we found only like 40 compared to like 2,500. That's a small number. So of SVs that can share by our method, most of the best would know. But if we only consider the homozygous SVs, then the PAVs we found is more than the traditional SV method. So in total, we think it's, our method is complementary to the traditional methods. Well, this all these data are deposited in the China National Center for Bioinformatics and also in the Shanghai branch, which called Node database. So here's my summary. We found about 80%, 80 million base pairs non-reference sequence, and about 1% of the genes are 1.3 something percent of the genes are dispensable genes or distributed genes. We found 4 genes that are more frequent in Chinese population, and at least 14 non-reference genes were found, and 1 were proved that can inhibit cancer cell growth. So this method is complement to the traditional method. That's all I have. And I want to mention one thing more is a high quality reference pangenome is needed based on our experience. And also based on our analysis on the third generation sequencing data, the non-reference genome, I mentioned it's about 1% to 10%, but maybe more. Because if we do analyze those genomes sequenced by third generation high quality genomes, then maybe we will have more or even much more novel sequences. Okay, here I will stop here.
Okay, thank you. Well, thank you for good presentation. Dr. Park.
Yeah, thank you for the great talk. And I just have a simple question. As you know, that regarding the human genome, the GRCH38 has some limitations on the coverage in the centromere region. So there are many missing gaps in the centromere region. And in your Chinese pangenome, did you figure out some novel sequences located in the centromere region? And also, do you have any plan to change the template sequences from HG38 to CHM13, which is recently announced by the T2T consortium?
Very good question. Okay, in the project I present now, we didn't focus on the regions in the like centromere with high repetitive regions. We did expand a little bit, not many. As you can see, as I mentioned that fully unaligned region is 30%, 30 million base, and the partially unaligned is about 50 million base pairs. So we did expand some of them, but not compared to the high quality T2T genome that was like 130 million base pairs more longer than the the human reference genome, right? So we did expand a little bit, not much.
And also, sorry for interruption. So, and also, do you have an experience? So, in the next plan, like
Yes, we are evaluating using the T2T genome as a reference versus the human genome reference as the base, and use the pangenome as a reference. And also we are testing the graphic model of the human pangenome as a reference.
Okay, thank you very much.
Any other questions? Okay, so thank you very much. Good talk, and I'd like to move the final talk from Ji-Hwan Park from COBIC. Are you ready?
Yeah, I'm ready. Can you see my slides? Oh,
yes.
Yeah.
Okay.
Let me start. So first, I appreciate the organizers of the ABC Symposium for giving me an opportunity to introduce the recent advances in the Korean National Genome Project. So my name is Ji-Hwan Park, working at Korean Bioinformation Center of Korea Research Institute of Bioscience and Biotechnology. So a drastic improvement in related technologies, including high throughput sequencing and data analysis and sharing, spurred several national and international genome projects. So all these projects made a new wave of the population genomics and precision medicine as an inevitable consequences. So national genome projects have been launched in more than 41 countries, including UK and USA, as well as the many other Asian countries. So our government also joined this new wave of population genomics and precision medicine through the Korean National Genome Project. So the mission of the National Genome Project, abbreviated as NGP, is to encourage scientific and industrial community to perform integrative analysis with the large-scale genome and clinical data while protecting the participants' privacy and securing their confidentiality. Therefore, our project enables application of precision medicine and diagnosis of diseases based on participants' consent. So let me briefly outline the workflow of the NGP. There are 3 steps in the workflow. The first one is the collection of samples and data processing and quality control, and data analysis and sharing. So there are 7 types of the cohorts in the NGP, and we collected whole genome sequencing data and clinical data from these cohorts. For rare disease cohort and COGES cohort, we newly generated whole genome sequencing in this project, and all these data are stored in the NGP consortium. After processing and quality control on the whole genome sequencing and clinical data by COBIC, the processed data are provided to the scientific community through the research environment platform. This platform provides computing resources as well, and the researchers perform integrative data analysis in the secured workspace. The National Genome Project, a pilot phase, provides genome and clinical information from up to 25,000 samples. So for rare disease, we collected about 15,000 participants, which consisting predominantly trio. And for normal participants, there are two types of the cohorts. The first one is COGES, and the second one is the KGP, as known as the Korea Genome Project by the Ulsan. And from those two cohorts, we collected the 5,000 participants and 2,383 participants. And for the currently incurable disease cohorts, consist of Alzheimer's disease and autism spectrum disorder. For cancer cohorts, we gathered colorectal cancer samples and non-smoking lung adenocarcinoma samples. So in those cancer cohorts, we both collected the genome sequencing data from the tumor and the paired adjacent normal tissues. So I briefly talk about the progress of the NGP pilot phase as of 1st November. So far, clinical data from 23,833 participants have been collected from the 7 cohorts. And we newly generated 20,000 whole genome sequencing data, and we also collected 5,000 whole genome sequencing data from NGP Core partnership. Here, the NGP Core partnership means the cohorts that are previously established by other programs in Korea, independent of our project. So this figure shows the timeline of the NGP from the beginning to 2022. So in January 2020, the Korea NGP has officially launched. So in the June of the 1st, rare disease patients enrolled and previously established genome projects have joined to the NGP. And this November, we finished data processing and quality control for 25,000 genome data and their corresponding clinical data. So by 2022, all these data will be open to the public. Now, I will concisely talk about the current issues about collection of samples and data. So first of all, we always be aware of our domestic law, named as the Bioethics and Biosafety Act. This law forces us to obtain the consent for participation. So regarding the process guided by the Act, the research participation agreements and plans are approved by multiple institutional review boards. Then blood or saliva samples and clinical information are collected according to the strictly managed quality control processes. So these are the so regarding the rare disease cohorts, these are the list of the hospitals that collect and manage rare disease cohorts. A total of 17 hospitals representing all the provinces of Korea joined the NGP. By collecting the data from the previously established cohorts, we collected diverse data sets involving normal patients, autism spectrum disorder, Alzheimer's disease, and colorectal cancer and non-smoking lung adenocarcinoma. We can save quite large amount of R&D budget from the NGP by collecting or by the NGP Core partnership. So in the future, research and medical staffs in NGP Core partnership can join the next phase of NGP, aiming to collect the data from more than 1 million participants. The NGP core partnership encompasses diverse types of cohorts, which includes cancer and currently incurable disease patients and normal healthy participants. From the core partnerships, we want to establish the standard operating procedures for diverse types of disease and healthy participants. The SOP involves ethical and legal and social implications, such as consent from the research plans regarding the prospective or retrospective studies in the NGP. Second, strategies for sample and data collection, for example, sampling and banking solid tumors rather than blood. Third, standardization or update of clinical information, for example, longitudinal analysis of clinical information and outcomes. The establishment of SOP involving these issues will be critical when we attempt to recruit about 1 million participants as the next phase of the NGP. So standardization and quality control of clinical information is an important issue in the NGP. So in the research environmental platform, we provides the structured clinical data. So for example, for 7 cohorts has a differently structured clinical data. So for example, rare disease cohort, they have human phenotype ontology, and for normal and healthy participant cohorts, the structured clinical data includes general health checkup, including interview, medical examination, radiography examination, blood test, and urinalysis. For the incurable disease cohorts, for Alzheimer's disease cohorts, it includes the blood test, urinalysis, education state, and mini mental state examination results. For autism spectrum disorder, ASD-specific information such as social communication questionnaire, autism diagnostic observation and schedule, and interview is included in the structured clinical data for ASD cohorts. For cancer cohorts, the clinical data include pathological diagnosis, metastasis, survival, medical treatment, and radio and chemotherapy information. So let me briefly talk about the genome sequencing step. So sequencing processes consist of 3 different steps: sample quality check, library preparation and quality check, and then sequencing. So for sequencing, in this project, we operated 13 units of Illumina Nova 6000, and the 13 units of the Nova 6000 can generate more than 41,000 samples annually. So all these whole genome sequencing data were generated by Macrogen, DNAlink, and Therazen Bio and Lab Genomics. So as of the 1st November 2022, a total of 20,000 samples were sequenced, and those samples came from 15,000 rare disease participants and 5,000 COGES participants respectively. Showing our outcomes as simple numbers, the sequencing depth of each sample was 138 gigabase on average. So these figures support the goodness of our genomic data quality. So the left figure shows the most of the bases in the whole genome sequencing data are more than Q30. Now I'd like to explain about our data processing and quality control processes. In collaboration with Seoul National University and Korea University, data processing and quality control pipelines for the whole genome sequencing data have been established to call germline or somatic variants with high consistency and confidence. Regarding the variant calling, last year we focused on single nucleotide variants and short indels, and this year we focused on structural variations, including copy number variation, translocation, and complex rearrangements. Also, we plan on performing a comparative analysis between our results and other public databases. This is the scheme of data processing and quality control pipeline for identification of single nucleotide variants and short indels. So to enhance data processing speed, we call germline variants by using the GATK Spark, which is 3 or 4 times faster than Java-based GATK pipeline. For somatic mutation, we applied FPGA-based Dragon platform, and this was 20 times faster than GATK Spark. So let me briefly introduce the research environment platform. We provide processed whole genome sequencing data. Whole genome sequencing data and the identified clinical information in this platform. The researchers can conduct a multitude of integrative data analysis in the secured workspace. So after variant annotation for the rare disease family, we could find some pathogenic or likely pathogenic variants. We return our data report to the medical staffs in the rare disease consortium, as I shown in the previous slide, and they finally diagnosed the diseases with these causative variants. So, however, we were able to diagnose only about 30% of the participants. Still, we need to further studies for diagnosis of the diseases and find the causative variants. So about 70% of the participants are still undiagnosed in the Korea NGP. Maybe some of them have structural variants that may affect the rare disease phenotypes. For those undiagnosed participants, we may need long-read sequencing data to achieve the high resolution to understand structural variants. So now I'll briefly introduce a spin-off project in COVID named as the Korean Pan-Genome Project. So since the Human Genome Project was successfully ended in 2003, there have been several international genome projects such as ENCODE and 1000 Genome Project, and these projects spurred the advancement in genomics field. But recently, there was an article in Science that announced the completion of human genome sequence by filling the missing gaps located in the 8% of human genome. So now we are fully aware of the importance of the human reference genome, such as GRCH38 and CHM13, that have drastically accelerated the human genomic study. So now what will be the next step in the genomics field? So including our findings in Korean National Genome Project, recent findings in a large amount of the population-specific structural variations implied the limitations of the linear reference genomes. So these suggest a graph-based pangenome as a promising alternative. So recently, the Human Pangenome Project is now working on it, and COVID also put many efforts on the construction of human pangenome. So to construct the graph-based Korean pangenome, it is preliminarily required to identify the uncharted structure variation that may significantly affect the structure of the graph-based pangenome. So, to briefly outline the workflow of the construction of the pangenome, first, performing de novo assembly of the long-lived sequencing data, and second, we can construct the complete reference genome using the long-lived sequencing data, and then the identification of SNPs, small indels, as well as the structural variations, we can incorporate this information to the pangenome. And finally, after the completion of the pangenome, we can use this pangenome to call diverse variants from the Korea NGP data. Currently, Korea Pangenome project is under the first phase. During the phase 1, we performed de novo assembly of the human whole genome sequencing data by using the PacBio Hi-Fi platform. So in summary, the Korean National Genome Project enables the researchers to explore clinical and genomic data from 23,833 participants corresponding to the 25,000 samples in the secure cloud service called Research Environment Platform. And second, COVID launched the Korean Pangenome Project as a spin-off project from the Korean National Genome Project to achieve the better performance, to identify the uncharted structural variations and integrate numerous genetic variants from Korean population. So first of all, I'd like to thank the major participants of this project, and many researchers in KRIB and COVID, including Director Sun-Young Kim, are involved in this project. And also, I'd like to thank KST, KD, KDCA, and NGP Core Partnership, as well as the Seoul National University and Korean University. And I'd like to inform the NGP supported by the 3 ministries in Korea, as well as the National Research Foundation of Korea. Thank you for your attention, and this is the end of my presentation, so I'm happy to answer your question if you have any.
Thank you for your great talk. And so is the Korean Pangenome project just getting started? Do you have any, already have some results from the pangenome?
Oh, so currently, for long-lived sequencing data, we use the 3 samples. So we made a template to make a template reference sequences. We only use the 3 long-lived sequencing data, and we will put the structure variation information and also SNPs and small indels from the 25,000 short-read sequencing-based genome data. So now we are in the phase 1. So we are currently applying several graph-based pangenome tools, such as the giraffe announced in Science last year, but still we have a long way to go. We should firstly we now benchmark several graph-based pangenome tools, and but in parallel we are now making have structural variation information from 25,000 samples. So we try to figure out how to incorporate this information to the pangenome. And so in our current study, we found that the server graph-based pangenome, their performance is quite similar. So, but maybe in the near future, we will select the one tool and after the benchmarking test, and then we will make a pangenome and we will open this result to the public next year. Yeah.
Right. Any other questions?
Yeah, yes. In the 25,000 genomes, how many of them are the third generation sequencing data?
None of them have been sequenced using the long-read sequencing data, but yeah, that's a critical question. But we actually for we used the independent three samples for the long-read sequencing data, and it's just independent of the 25,000 short-read sequencing based data, because we tried to make unbiased templates. So that's the reason why we generate the long-read sequencing data from the independent participants.
Okay. Yeah, I think the human pangenome is going to be very important. Well, help study. Yeah.
Right.
So did you join the human pangenome consortium?
Not yet, but maybe actually this should be considered by our director after some Kim. Yeah.
Okay.
Yes, actually, this discussion with PacBio team members to join the Pangenome Consortium, but as we don't have big grant, we still want to get more money to join that community.
Any other questions? If not, I'd like to close this session. Thank you, Dr. Park. So, it was again a very short symposium this year, and but although we are unable to meet face to face again, but it was good to hear each other's updates and high-level science talks. And so, I hope to see each other again